Let me start with a bit of background. Neurodevelopmental disorders are mainly characterized by delay or disturbance in the functionality of the neurological system. They tend to show a heterogeneous phenotype, and some of these disorders are relatively common. For example, 1 in every 54 children in the US is in the autism spectrum. Also, we observe high comorbidity among these disorders. Among all neurodevelopmental disorders, autism and intellectual disability are one of the most comorbid pairs, having the largest effect size for shared genetic risk. General symptoms for ASD are restrictive and repetitive behaviors, dysfunctional reciprocal social behavior, and impaired communication abilities. Meanwhile, ILD is characterized by below average intellectual functioning, in other words, an IQ score less than 70. Both are relatively common in the population, and we observe that 70% of children with ASD are also diagnosed with ID. They show similarity in their genetic background, as both disorders are affected from CMVs and single gene mutations, and both show oligogenic mode of inheritance. For autism, the genetic circuitry is like a large puzzle. Currently, we know approximately 100 genes, which refers to 10% of all pieces. It is costly to experimentally pinpoint all genes. However, we also know that these pieces are interacting with each other in biochemical networks, and we would like to use these interactions for our sake. So we try to answer, can we use the goodbye association principle to pinpoint connecting pieces? Even though the performances of earlier studies are noteworthy, all methods suffer from some shortcomings. Firstly, by design, they only work on a single disorder. To work on comorbid disorders, they may act as if both disorders are the same and back their mutational burden. But then the disorder-specific features are lost. As another way, they can perform independent analysis, then combine their results. But this time, the information coming from shared genetic architecture is ignored. Secondly, they work with at most a handful of networks, and they perform additional analysis to figure out where the actual signal is coming. Then it is difficult to deconvolve the results. For example, in a study published on Cell in 2013, they need to do a downstream analysis to find out which brain region clusters has higher significance at which neurodevelopmental windows. We would like the method itself learn and make use of the significance of these networks. So we observed these shortcomings and we propose a new method which overcomes them. Our method is named DeepND, which stands for Deep Neurodevelopmental Disorders. The model uses a multitask learning technique to analyze comorbid disorders. Here we have a multi-layer perceptron to process common features. After that, graph convolutional network layers process multiple networks simultaneously. We also incorporated the mixture of experts model to indicate which networks that it utilizes are more informative. The darker the color, the informative the network, and larger the weight assigned to it. Here are our results. We run our model in two setups. One is the single disorder version shown as DeepND-SD, and the second one is the multitask model. We compare our results with Krishnan et al.'s weighted SVM in terms of area under ROC curve on the left and area under PR curve on the right. For the sake of fairness, we run the SVM with our new ground truth sets to compare. In this figure, the gray box plots are results from the weighted SVM, and the purple boxes correspond to the results of DeepND in single task setting. Finally, the pink boxes show the results for DeepND in the multitask setting. The single task setup outperforms the state of the art in 3 out of 4 comparisons. For both disorders, the multitask approach surpasses the performance of all single task methods. We also share our results in terms of precision recall curves. In this figure, E1 is the set of highest risk genes, and E2 is the set of second highest risk genes. Here again, DeepND performs best among all other methods for both disorders and both risk gene sets. By examining the results from mixture of experts module, we could also create heat maps for each simultaneously processed disorder to see which brain regions and time windows are more informative. DeepND assigns higher importance to early fetal to mid-fetal for PFC-MSC brain region cluster. This is in line with the findings of Wilsey et al. However, DeepND generally indicates lower informativeness for MBCBC cluster. In conclusion, DeepND is the first multitask gene risk discovery algorithm which can work on comorbid disorders and can utilize multiple networks and deconvolve the informativeness of each gene interaction network considered. It also can be generalized to work with any combination of disorders and diseases with shared genetic architectures. It helps dissecting out ASD and ID-specific genes by predicting several novel genes. Before finishing, I would like to thank my co-authors, our supervisor and mentor Ercüment Çek, and my colleague Oğuzhan Karakahya. You can use the QR code on the bottom left to read the full text. Thank you for listening.